video. Hello, so this
Hello.
Hey, what's up?
Hey. All right.
I can't find my Zoom screen for some reason.
Oh, that's peculiar.
I can't find my Zoom screen. Oh, got it. It was minimized. Okay. Oh, where are you?
Me?
Mm-hmm.
Oh, new room.
New room in the same
Yeah.
apartment?
Yes.
Do you change with like your mom or something?
What happened?
Do you change with your mom?
Yeah.
Okay, cool. Looks really nice. Oh, okay. I got the PowerPoint. Okay, where do you want to start?
Over here, chapter 1. Let's see. You disable my screen sharing.
Oh, yeah. Here, one sec. Okay.
All right.
Cells, the fundamental units of life. Okay. Okay. All right. Do you know what a nucleolus is?
One nucleolus?
Oh, yeah. Mm-hmm.
Yeah, it's like, what do you call it? Isn't it part of the nucleus that stores RNA?
Yeah, it makes a ribosomal RNA.
Yeah, ribosomal RNA. There you go.
Yeah. Because there's ribosome, yeah, inside of the nucleus as well. Okay, and then cytoskeletal stuff. All right, we can move on to the next one. All right, you pretty much know what the cytosol is.
Cytosol.
Mm-hmm.
Cytoplasm equals formulas. Oh, that's just like the, what do you call it? The membrane spacing, right?
So what's up?
That's like the membrane spacing, right?
Membrane spacing?
Spacing.
Yeah. What do you mean by that?
Like, that's what cytosol is, right? Just trying to remember again.
Oh, it's just literally what's in the cell. It's pretty much like water.
Oh, okay.
Yeah, it's like what's inside. Usually they would talk about cytoplasm, and the cytoplasm is just the cytosol, including the organelles as well.
Okay, so cytosol is organelles.
Oh no, cytosol is just the like stuff that's inside of the cell, and cytoplasm is the cytosol in addition to the organelles, so pretty much everything inside.
Oh, okay. So cytoplasm is just everything inside, right?
Yeah, cytosol plus the organelles, yeah. Yeah, so in that diagram, essentially everything on the right side, like B, everything is like that bluish color or whatever.
Mm-hmm.
So see how that isn't including the organelles? So that's
Yeah.
just the cytosol, yeah.
Got it. Oh, whoops. There you go.
Yeah.
Helix cells.
Yeah. The immortal cell line.
Yeah.
Yep.
Okay. And then much of what we know about cell bio comes from cultured cells.
Yep. That's her.
Okay.
Alright, and you know about this, atoms, right?
Mm-hmm.
Yep.
Atom.
So yeah, basically the outermost electrons, otherwise known as valence electrons, determine how atoms interact with each other.
Mm-hmm.
Yep, most stable when its outermost electron shell is filled. If it's unfilled, it's going to be reactive.
Okay.
Yeah. Alright, covalent bonds, ionic bonds. So, you know that as well.
Yeah. Yeah. Transfer an electron for an ionic bond.
Okay. We pretty much know this stuff.
Mm-hmm.
Yep. We know that as well. Yep, they're strong.
Yeah.
And this is water. So, it's that bent shape. And note that symbol is a lowercase delta.
Mm-hmm.
And it indicates partial charge.
Indicates partial charge. Okay.
Yeah. So we see partial negative charge is towards the oxygen, partial positive is towards the hydrogens, and so it has a dipole moment.
Mm-hmm. Mm.
Yep.
Mm. Mm. Mm.
Yeah, I looked through the
Oh.
slides. Most of these first ones are going to be stuff that we know.
Mm-hmm.
Non-covalent, essentially, are just intramolecular forces.
Mm. Mm.
Yep. Okay, so they'll talk more about this later, where they in this PowerPoint, where they talk about this. So we can kind of like go through
Mm-hmm.
this pretty fast, like the first couple. Like next, they're gonna show you the types of intramolecular bonds.
Oh
Um,
yeah, that's the.
mm-hmm.
Um.
Yeah, even he's like, I'm not gonna talk about this because you should, you already know it.
Yeah. Mm-hmm. Slides and these topics in the block for the region.
Yeah. So hydrogen bonds are the strongest type, and here's an example of them forming between water, right?
Yeah.
And then there's an example of them forming with sugar.
Got it. Mm.
Okay, so this is kind of like a close-up view of it, and it's just going to be something like with a partial negative end is going to create a hydrogen bond with a partial positive end. So as you
Mm-hmm.
can see, there's a double bond on the left between the CO. And so the oxygen, which is the more electronegative of the two, has that partial negative charge. And on the right, you have this amine over here with an NH, and the hydrogen has a partial positive charge. So that's why there's a hydrogen bond between the two.
Got it.
Yeah. All right, so now we're going to talk about polymerization of, molecules, like biomolecules. All right, so sugars combine to make polysaccharides. Amino acids combine to make polypeptides, or proteins, right? Nucleotides combine to make nucleic acid. Now, when you synthesize, it's a condensation reaction, otherwise known as what?
Dehydration synthesis.
Exactly, because you create a water by doing
Mm-hmm.
that, right? And then the reverse, you have to add water, and that's hydrolysis.
Yeah.
Yep.
Okay. Just making sure. Okay. Um, So just making sure hydrolysis, you're adding water, condensation, losing water, right?
Yep.
Mm. Okay.
Yep. So here's just an example. Subunits make the macromolecules, which then turn into larger things.
Covalent bonds.
Mm-hmm.
Oh, yeah. Subunits turn into covalent, macromolecules turn into non-covalent bonds.
Yeah, like proteins and things like that are going to be held by intermolecular forces after that point. They'll talk more about it when they, like later in this PowerPoint, when they talk about primary, secondary structures.
Oh, yeah. Primary, secondary, tertiary, and quaternary.
Mm-hmm.
All right.
Yeah.
Okay.
So we can just go, yeah.
All right.
Because they talk about it more in depth later in the same PowerPoint.
Got it. Carbon. So 4 types of biological macromolecules.
Yeah. You know that stuff as well.
Yeah. Yeah. I'm just, I put them in notes in here just in case.
Mm-hmm.
Sorry. Because it's like new, it's like new subject, so I make sure I have it all in one place.
Oh yeah. I can tell you like what would be
Good
like,
to write down, right?
Yeah, what like the important notes are.
Okay. Got it. Okay.
Okay. So here I'm gonna try to like log into Zoom on my tablet as well, because Zoom has like this annotation feature, which is actually pretty helpful, and I'm gonna see if I can use that to write. But all right, now I have to let myself in. Okay. Okay.
Okay.
Oh, God. All
Yeah,
right,
just like
awesome.
turn off the mic on one of them.
Hmm?
Turn off the mic on one of them.
Uh, sorry.
I guess on the tab we'll like turn off the mic and then turn off the sound.
Test. Man, I hit mute. Mute.
Yeah, you muted the tablet. That's good. And then just gotta mute the
Test.
sound off your
Test.
tablet.
Got it.
Got it. All right, good.
All right. Oh, awesome. This works. Okay. All right. So I would write something like this. Oh wait, what? Oh. Something like that.
Okay.
Yeah, I wanted to do this because I think we should start with your because with a lot of like my MCAT students, like they have a hard time writing like notes and shorthand when it comes to like the test and stuff like that. And so like I want to like have you start learning how to like take notes in like kind of like effective like shorthand.
Yeah.
So you see how I wrote like these like greater than signs?
Yeah.
And then underneath I wrote essentially the number of sugars that they're composed of. So if it's less than 3, they'll be considered monosaccharides. Actually, oh wait, it doesn't let me erase in the same way. Yeah, that's even better. Now,
Okay.
let me ask you this. Why is it better? It's better because if we know that oligosaccharides are 3 to 10, we know that
know it's a
monosaccharides
short
must
walk.
be less than 3, and poly must be greater than 10, right?
Yeah.
So we don't have to write that extra stuff.
Oh, not being like short,
Yep.
small, or long, because we'll be writing now.
Yeah. And like this type of like shorthand is like I want to like have you start with that like as soon as possible.
Okay.
Yeah. Okay. Now, chemical energy. All right. Stored in covalent bonds. Sugar polymers are the most abundant structural components on Earth.
Should I write any of that?
Yeah. Let's see it. Let's see if I can write. Oh, cool. I can draw there too. Yeah. So whatever you have for carbohydrates, just write like primary source of energy and most abundant.
What's the primary source of energy?
Oh, so this is all under carbohydrates. So I would write, in your notes like carbohydrates.
Oh yeah, I put carbs and then forms with saccharide and brackets and then mono. And then what you wrote there.
Yeah.
So then I put in star next. So I put primary source of energy.
Yeah, I would put like 2 bullet points, like primary source of energy, like for the cell,
Mm-hmm.
And it's the most abundant as well.
Okay. Most abundant structure. There you go. Oh, it keeps the notes from the last one. Oh, okay, cool. Structure of monosaccharide simple sugars. Okay. Glucose is a key molecule in the metabolism of plants and animals.
Yep.
Okay. That's not really crazy important.
All right, and that's just an example of condensation reaction right there.
Uh-huh. Yeah, I lost.
So you could write let's see. Glucose polymerizes to glycogen.
Okay. I'll still be putting this under carbs, right? Yeah.
Yeah, I'll tell you when to. Yeah.
Okay.
Yeah.
Glucose polymerizes to glycogen. Nice. All right. So the glycogen is basically no, what do you call it, H2O. Missing a H2O group.
Yeah, because it's a dehydration.
Yeah. Got it. Okay. Galactose. Sounds like a Marvel villain.
Wait, what? Oh, like galactose?
Yeah.
Yeah, that makes lactose. So yeah, these are two sugar
I think
units.
that's the right time, right?
You know, it's like I have like my own notes on these biomolecules that are even better, frankly. That I can share with you. Like, do you want to do? Wait, let's see. We can do something like, I mean, I have these same PowerPoint slides open, and you can tell me like how to go like through them. But I kind of also want to show you like here, like I never got it like the having both of us share our screen to work out. Because I think you need two screens, so I'm gonna try that out. I'm gonna go do this and do that. Now can you see my shared screen? Do you see my shared screen?
No, it's still you on the camera.
Okay, let's see. You can still see only... You can't see it still?
No, I still can't. I still see you.
Okay. Alright, like, hit stop share for a sec. I just want to show you.
Oh, you can share two screens at once using the shift button.
Yeah, I tried that. Wait, I tried doing that. Alright, let me try it again.
Well, maybe because I need to stop sharing. Hold on.
Yeah, but I have it so multiple participants can share simultaneously, but I don't know why that hasn't been working out.
Oh, okay.
But I think it... Yeah, it says dual monitors recommended. Do you have dual monitors, or...
I have my laptop screen and then my monitor here.
Okay.
So I can like put my, what do you call it? What do you call it? This on my little screen here. Like that. And then I can have you over here.
Alright, I'm going to try then. You see anything? Yeah, do you see my screen?
Huh?
Yeah?
Your screen?
Yeah, do you see it?
No.
Damn it. Okay. Because I could see your screen on my tablet, but I can't on... Oh, but you can't see mine? Okay, so try stopping your share for a sec, and you should kind of see it now.
Yeah, I see yours now.
Alright, yeah, so check this out. This is like basically even better like notes because I put everything together. So like maybe you can write, you know, this down in your notes.
Yeah.
And I also have, you know, like the PowerPoint here.
Okay. I mean, I can load the PowerPoint over here too on my thing.
Yeah. But then we'd have to like go back and forth. So I guess I can just like go over this, like, I guess pretty quickly. Just I want to have you write down a couple of things. Like for instance, like this is true for everything. All polymerization are dehydration synthesis, which equals condensation, and it requires energy output input.
Okay. Got it. And this still goes under carbs, right?
This goes for everything.
Oh, okay.
So like that's why essentially like I have like the notes, like, you know, for essentially for everything in this manner.
By the way, you left it. Hold on. I'm still looking.
Oh yeah, yeah.
Energy input?
Yes.
Cool.
Yeah. So you see how I wrote like all underlined polymerization are dehydration synthesis underline equals condensation, and then this big arrow requires energy input.
Mm-hmm.
Now, I didn't write this because it's like, you know, like if this is true, then the other thing is true as well. But like, you know, if you wanted to know, I can write it down in the annotation thing. But check this out. So, all hydrolysis. Whoops. Oh man, I hate how they don't let me use the back. But I'm trying to think of breaking down the opposite of polymerization. So all, like, let's just say, breaking down are hydrolysis reactions, and guess what? They do not require energy, but in fact they have energy output, meaning they give off energy.
Oh, okay. So what does that word say after all?
Oh, sorry. Because yeah, this thing is, remember that it like bends.
Oh yeah.
But I was trying to think of the word for the opposite of polymerization, so I just wrote breaking down.
Oh,
But
okay.
you could use whatever you think would be better.
And then equals arrow gives off energy.
Yes. All right. Did you see the new thing I wrote?
Yeah.
So when the sugar has 5 or more carbons,
Uh-huh.
it prefers the ring form.
Okay.
So essentially, I wrote out bless you. So like this is like
Yeah.
a Fisher projection of a sugar,
Okay.
and essentially like you draw it like this line where, and then these wedges mean that it's going like into the paper, and then these black wedges means it's pointing out towards us. And so that means that, I redrew it here. And so like, I numbered the carbons like 1, 2, 3, 4, 5, and 6. And this over here is, I think, glucose. And essentially what happens is this hydroxyl group attacks that, and it closes to form a ring. So like in the PowerPoint, it was just showing us the ring forms, but it didn't really tell us this additional kind of information.
The why.
Yeah. So
Okay.
the why is because, this hydroxyl group essentially, interacts and bonds with the carbonyl group over there. Because if you remember, the carbonyl group, right, has a partial positive here, partial negative here, right?
Yeah.
And an OH group has a partial negative, right?
Yeah.
So it attacks like that.
Read your way into that. Oh, you left it.
What's up?
You left it a second.
Oh yeah, I realized that. So I actually like talk about it here. So this is what happens. The OH on the 5th carbon, nucleophilically attacks the carbonyl. So this is what happens. The arrows here to make a 5 carbon ring, and so we have 6 carbon ring over there, and this is called a chair. This type of drawing.
Yeah.
So it's essentially a 6 carbon ring like this, but if you imagine it in 3 dimensions, like imagine this part bending upwards and this part like tipping down, it looks like that. Yeah. So that's a chair, it's called. And so this is just an example of what I was saying about how it prefers the ring formation. Because the straight chain version of glucose exists less than 0.1% of the time. Right?
Okay.
Now, so now there's in this type of in a chair there's going to be basically 2 types of positions things can be. That it can be equatorial, which means that it's in the same axis as this chair molecule, or it can be axial, which means that it's just going straight down instead of being in the same axis. And basically, if something's equatorial, it's more stable that way. So, you don't have to know too much about equatorial or axial, but essentially you see that this version of the ring is 64% of the time. Like, if you just have any glucose molecule, it's going to be, it's called mutarotation. It's going to be going back and forth between these types. And 64% of the time, it'll be this type of ring. 36% of the time, it'll be this type of ring. But essentially, it's always going to be a ring. Like, because straight chain form, it'll just be like that for 0.1%.
Okay. What was that thing you wrote in green there?
This?
Yeah.
Oh, this is just, I didn't know if I should go into it more in depth because it's kind of an orgo thing. But
okay.
It talks about, but essentially what is important here is just how I kind of drew it before, but essentially there's a partial positive, partial negative here, and oxygen on the hydroxyl group has a partial negative charge, so it acts as something called a nucleophile, which means that it can attack this partial positive charge, causing it to make a bond with it.
Okay.
Yeah, you'll see that a lot in organic chemistry.
Mm-hmm.
Yeah, but that's essentially what happens. I mean, it's
Okay.
if you wanted to know, that's also why RNA is so unstable compared to DNA, because DNA is deoxy, right? RNA
Mm-hmm.
has an extra OH group, and OH groups are nucleophilic, so the RNA's OH group is going to attack itself.
Okay. So that says the OH on C5 nucleophilically attacks with carbonyl on C1.
Yeah, that's to make a 5-carbon ring. Now, if it was making a 6-carbon ring, it would be this one that attacks.
Number 4.
No, 6.
6. Oh,
Yeah,
okay.
this guy over here.
Mm-hmm. I see.
Mm-hmm.
So OH on C5.
Yeah. So see how it's the O that hits that carbon? So that's why you have this bond over here. In this ring, you have that oxygen in that ring.
Yeah.
Yeah.
And then if it's making a 6-ring one,
Yeah, what about it?
if it's making the 6-ring one, it's going to be the 6 that attacks the.
Yeah, this one?
Oh. Uh-huh.
Mm-hmm.
It'll attack the carbonyl at C1.
Yeah. It'll always attack the carbonyl at C1 because it's a carbonyl.
Mm-hmm. That's the H2O2 nucleophilically attacks. Carbonyl at C1. To make 6-ring. And then for this one, it's 5-ring. Cool. All right, got it.
Yeah. Now I'm looking for. One sec. What? Running out of time? That's weird.
Running out of time.
Yeah, I've had that happen before. It's just, we could just restart it again. But I was looking for another one where I was, where I go.
It's gonna be like it's barely been an hour.
Yeah, it's just a way for them to make you want to, whatever, buy more
the
crap.
premium version.
Yeah. There, one sec. I was looking for, because when the glycogen thing happened, I had like this awesome diagram of the glycogen. I just don't know Where? No, that's not it. I put it, but okay, whatever. Until I find it, I could just go over these, like where we left off.
Yeah. We left off at disaccharides.
Oh yeah. So these are glycosidic bonds that form, and so sucrose is an example of glucose and fructose combining, and lactose is glucose and galactose. Make sense?
Yeah.
Polysaccharides, many sugar units, important for energy storage and cell structure. So, yeah, this is the glucose thing that I was trying to freaking find. I had like a really cool kind of note for it. But whatever. So, yeah. Lipids. So now you can start in your notes talking about lipids. So I have that. Oops. So I have that. Wait, I guess not there. Where the hell? Oh, wait. Damn it. Okay, so anyway, forms diverse structures.
Yeah.
Oh, got it. Okay, here. Okay. So, here. This is basically lipids right here. Put that there. There. Can you see both of these? Okay. So you see how it says diverse structures, fats, steroids, phospholipids, etc. And then the functions.
Yeah.
So I talk about that more in depth over here. So essentially, you can write like lipids. So you can say, since they're only saying fats and steroids and phospholipids, you can essentially just write, like, for lipids, those ones instead of all the ones that I have. But for instance, you can like see like the structure. So you can start by this fat structure over here.
Okay.
Because you see in the PowerPoint, they're talking about it right here.
Yeah.
Yeah.
Triglycerols with 3 fatty acids connected to 3 hydroxyl group, right?
So yeah. Wait. Yeah. That's what glycerol is, correct?
Okay.
And then it'll combine with fatty acids to make the fat.
Okay.
Yeah, so I have that like over here. Okay, and fatty acids have a hydrophobic part, which is just a bunch of carbons, and the acidic part, the carboxylic acid part over here, which is hydrophilic.
Yeah.
So it has a hydrophobic part and a hydrophilic part.
All right, so wrote.
Yeah, what's up?
Okay, so I wrote the forms. Next part is what?
What do you say about the forms?
Oh, no, I wrote down what the forms were: the fats, steroids, and phospholipids.
Oh yeah,
stuff.
yeah.
So what's next I should write down?
Yeah. So under the forms, we're dealing with just the fats right now. So like in your notes, you could write like lipids. So like fats, and now we're gonna write stuff about fats, and when we're done with that, we're gonna write stuff about phospholipids. Right? We'll write some stuff about that, and then we'll end with steroids. And, as we go through that, I'm going to show you over here, like, phospholipid over here, and then, like, steroid over here. Whoops. So, Yeah. Oh, damn it. Yeah.
All right.
Yeah, I don't know why it does that. It's annoying.
Mm-hmm.
All right, so we left off here. So, so for the fatty acid part, I want you to distinguish between saturated and unsaturated. So, in your notes, you can do like, for the fatty acids, right? There's saturated and unsaturated. So they're referring to saturation, like essentially, in terms of like saturated with hydrogen. So in other words, saturated means it's just full of single bonds, and unsaturated means that there's a double bond involved.
Got it.
All right.
Okay.
Now the double bond, what that does is it creates like a kink in the structure. And that means that it cannot be compacted as tightly.
Okay.
So in other words, yeah.
All right. So saturated refers to what?
Saturated. Okay. So saturated means. Whoops. Forget that. Damn it. Saturated means single bonds only. And what that means is that it can let's see. Can compress all the way to being a solid. So an example of that is butter, which is a solid at room temperature.
Double bond creates what?
Okay, so I wrote single bonds only can compress all the way to a solid. So an example of that is butter, which is a solid at room temperature. Now, unsaturated, they have a double bond. And what that does is it creates like a kink, K-I-N-K. And it cannot compress all the way. So that's why olive oil, which is an example of that, is liquid at room temperature. Yeah. And also, like, that's why, they say like saturated fats are bad for you. It's bad for you because it can help create those plaques in your arteries, the solid, things that clog up your arteries. Yeah.
Yeah, the saturated fats, right?
Yeah, and if you're wondering, trans fats, they banned those like a decade ago. That's just essentially using this like organic chem thing, like a reaction to take a double bond and to turn it into a single bond. So that's bad too. Make sense?
Got it, yeah.
Okay. And yeah, here's, another example of a condensation reaction. So we have the glycerol here, we have the 3 fatty acids here, and through condensation they make this ester linkage. And yeah, that can be 3 identical or different fatty acids.
Okay.
All right. Now, I just want to say this. If this was a phosphate, right, then you would get a phospholipid.
Oh, okay. I see.
Yeah. So I have that right here. You see that?
Mm-hmm.
So, like, this is a regular, fatty acid, phospholipid right here. And since it's charged, it's going to be hydrophilic.
Got it.
So that's why, it's in the lipid membrane. All right. So this is exactly what I said. Double bonds make kinks. The more double bonds, the more liquid at room temperature. Animal fats tend to be saturated. Plant fats tend to be unsaturated and liquid like oil. Hydrogenate vegetable fat to make solid margarine. So that's an example of trans.
Mm-hmm.
Storage form in plants and animals. So yeah.
Got it.
Mm-hmm. Again, saturated and unsaturated with a double bond. So let me talk about what cis and trans means. So when it comes to a double bond between, two carbons, right? Let's draw a line down that double bond. And now where things are, and it's funny because I just had this question with my student yesterday. It was an MCAT question, but it basically, so like something that, or you take the highest priority group that's left of the double bond and the highest priority group that's on the right of the double bond. And if they're on the same side, it's cis. If they're on different sides, it's trans. So for example, if I have something like this, like, something like here, like whatever, I'll call it like hydrogen, and then here, hydrogen, are these, so that this over here is going to be trans because they're on opposite ends.
Okay.
Now, if it was like this, this color, you can't really see much. Let me see. If it was like this, it's cis. They're on the same side.
Okay. So just give me a second.
Yeah, inorganic, so like here, trans, cis. That's also now where like the terms that are popular now in terms of like gender identity, cis and trans, comes from.
Mm-hmm.
Yeah. Yeah, and in organic chemistry, when it comes to naming compounds, you actually use German words for the two. So cis is this thing called Zusammen, and trans is a word called entgegen. Oops, yeah.
Okay. And then cis can be translated into a what type of acid is that? Like?
Oh, yeah. So that's just an example of a cis one, and we don't really need to know that much about. I mean, yeah, that's olive oil essentially.
Okay.
Oh yeah, I guess what's important is that the cis ones create like, like the kink. Like, well, they both create kinks, but they create a kink that has a bigger. I'll write bigger kink because if you just look at these two, like for this over here, that double bond doesn't really affect that overall structure, right? It's still this straight line over here.
Yeah, like it creates a kink, but it doesn't do much.
Exactly. Whereas this cis kink, and again, do you see how over here they're on the same side of that double bond? Like here it's going this way, and here it's going this way, same side. Whereas here, right, this carbon's going like up, and this going down. So that's trans. So the cis ones create like a kink that puts like a bend in the entire, like, you know, molecule. So trans fats is like this. It's where they take some, you know, hydrogenated vegetable oil. Like they basically take it and they put, you know, a trans double bond in it so it doesn't make that kink. That's why it's called trans fats. Remember trans fats? Yep.
Yeah, they're like really bad for you, no?
Yeah, because, you know, it makes it easy to turn into, you know, a... So this is what they say, hydrogenate vegetable fat to make solid margarine. Yeah, so essentially it's bad for you because it can be compacted into a solid. And, you know, therefore it can clog up your arteries with plaques.
Yeah.
Yep.
Got it.
And now, so under lipids you have like number one, like, you know, fats. And then the second category is phospholipids.
Yeah.
Yeah, so that's, you know, essentially glycerol plus 2 fatty acids and then a phosphate group. Okay? And the phosphate group is bound to a polar group. So in this case, you know, it's choline. But essentially, that, you know, it'll have a polar end to it. So that's why, you know, when you draw the lipid bilayer, right, we draw it like that. So you see how these two tails are these, you know, tails over here, and then this polar part is that right there.
Yeah.
Okay. Oh, and amphipathic just means that it can behave as both, like, whatever, hydrophobic and hydrophilic. Yep, and then that's exactly what we have here. And, you know, this is a good thing to draw, actually, because it kind of differentiates the glycerol from the phosphate from the polar head group. So, like, you know, if I were to draw it, I would just do like this, like, glycerol, phosphate, polar group, and then, you know, like fatty acid, and then you can do something like, you know, hydrophobic, hydrophilic, philic.
So should I write for phospholipids to start off with before I draw that?
Oh, just that it's this kind of structure, like when I went over it here, like see that?
Mm-hmm.
So it's essentially just it's the same thing as a fat. So if you drew like a structure like that for the fat, you can draw one for a phospholipid, but it's just it's just gonna have that phosphate group. So you can essentially write. I mean, you could essentially just draw this picture because it's really just these two pictures are the same, but this really makes it easier. And you can write down like here what I drew, and then you can also add, it's essentially I'll put this on the right. So a phospholipid is essentially glycerol plus 2 fatty acids plus a phosphate plus a polar group. So like here they wrote glycerol plus 2 fatty acids plus a polar group bound to a phosphate group bound to a polar group.
Okay.
Yeah. So like you could draw like these things that I wrote.
Okay. All right.
Okay, now for steroids, all they have is this slide, but I have the perfect thing for you to write for steroids. So this right here. So essentially, so this structure over here, I would draw, and once you have that drawn, yeah, tell me once you have it drawn, then I'll go over this thing.
All right, so draw that structure.
Mm-hmm. Are you still drawing the structure, or?
Almost done. Yeah.
Okay.
Just messed up a second. Now I'm done, yeah.
Okay.
Sorry.
So now this is like this over here is pretty much like golden, like essentially everything you need to know about this. And you can write it in the same, like after I explain it, you can write it, and you can write it in the same way. Like you see how I use like black to make the pathways, and then green to mean like, where it's happening, where it's entering and stuff, and then blue for the types of things that they are. So essentially, like I'll just like explain it first. So everything that's a steroid or a sterol, it's given that name because it comes from cholesterol. Okay?
Mm-hmm.
Now, cholesterol basically has this structure, this 4-ring structure. Okay?
Yeah.
Now, so okay, yeah. Before you, do you want me to like go over it first, and then you can write down this stuff?
You can go, yeah, start going through it while I'm just looking, yeah.
Yeah. So cholesterol is synthesized in the cytosol, all right, and then ACTH is a adrenocorticotropic hormone. When that is released, it causes the cholesterol to enter into the mitochondria, where it turns into something called pregnenolone.
Do men also produce that? Sounds like something that women produce only.
Oh yeah, we haven't gotten there yet. This is for everyone.
Oh, wow, okay.
Yeah. So pregnenolone is formed, and then it enters the endoplasmic reticulum, where it produces progesterone. Now, from that point, like progesterone obviously is for it's something that is important for women, but men also produce it. It's just that it doesn't stay as progesterone. So progesterone, from that point, it can turn into several things. Okay, it can turn into the stress hormone cortisol. Like you've heard of cortisol, right?
Mm-hmm.
That like goes up when you're under stress, and
Yeah.
that is a type of glucocorticoid.
Okay.
Which kind of gives you a clue. That means it's, there's some type of like sugar attached to it. All right. Now it can also turn into aldosterone, which is a mineral corticoid, because that works essentially to like have your whatever body like reabsorb salts and stuff. Mm-hmm. Now if it goes into the gonads, okay, it'll turn into testosterone, which is an androgen, meaning, it's going to produce male characteristics. And testosterone can turn into estradiol or estrogen.
Okay.
And this is the same for everyone. It doesn't matter if it's a male or a female.
Mm-hmm.
Yeah.
Okay.
So this is like the entire, like, for every single steroid, in addition to where it comes from and where these things take place. So this is basically, like everything you need to know. So some people who like abuse like anabolic steroids, they get something called gynecomastia. Have you ever heard of that?
Yes.
Okay.
It's when too much estrogen starts to get created.
Yeah, because
for men in it.
Because estrogen is created from testosterone. So if you have an excess amount of testosterone, that you inject, then that testosterone aromatizes into estrogen.
Yeah.
So that's why they need to take like an inhibitor of an enzyme that would aromatize it. I used to have like pregnenolone as a supplement
Mm-hmm.
because it's supposed to be like good for your brain.
Ah, okay.
And like a couple of other things.
Anagen, this first one?
What's up, androgen?
Androgen.
Yeah, andro means like male. So like androgynous, means like looks kind of like a boy.
Mm-hmm. All right, just 1 second.
Yeah, pregnenolone is cool because it acts as a neurosteroid actually.
Really?
Yep. So it can like cause like things to grow in your brain.
Oh, wow. That's not a good thing.
Oh, like not like to the extent of it being cancerous.
Oh, okay.
Like I'm reading right now, it was found to induce tubule polymerization in neuronal culture to increase neurite growth.
So cholesterol synthesized in the cytosol?
Oh yeah. Mm-hmm. Yeah, it's important because like a lot of people when they think of steroids, they only think of what people
Nucleus?
take to like
Oh.
bodybuilders take. They
Mm-hmm.
don't even realize it's just testosterone, and they don't realize that steroids is also like you could take like a steroid medication for like a billion things, right? Oh my god, why is
Yeah.
it giving me the 10-minute thing again? All right, that's really annoying. Um, yeah. So, but like steroids includes basically all of this stuff. It just means that it's made from cholesterol.
Cool.
Yeah.
Okay. Well, the good thing about the 40-minute thing is that it gives us like a time thing, basically.
A time thing?
So we know how we're doing on time.
Oh yeah.
Because basically we go through three, if we go through 3 of these and stuff, we still have an hour left.
Yeah.
So we know that there's like another hour
Mm-hmm.
to do.
Yeah. Yeah, that's so annoying. Okay.
Yeah.
Because sometimes it doesn't do it, but sometimes it does. Yeah, I don't know. Anyway, so yeah, steroids. So, that's all they have for it.
Mm-hmm.
I guess the only thing that you can add that I didn't have was that it's a component of the membrane.
Okay. Okay. What's next?
I think I know what might be causing this, but I'll fix it on the next one. But I think it's because I'm here. Here. All right. So the next time it shouldn't give it to us. I think it's because I signed on. There's 3 people technically because I signed on using my tablet.
Mm-hmm.
Yeah. Okay. So nucleic acids are the next ones.
Okay.
What are you up to?
No, it's just answering emails for quick from a teacher.
Okay.
Okay, sorry. Next.
All right. Yeah. It's just nucleic acids. So
Okay.
essentially that, and nucleotides are the monomers for it.
Nucleic acids. Forms. Okay.
Okay. I'm gonna test something out. Stay in here. Yeah, stay in the
Okay.
Zoom. I'm gonna try something. Okay? I'm gonna try because I looked up something and this might work. So I'm gonna try something. Okay? I'll be right back. But stay in the
Okay.
Zoom. Damn it. Yeah. Oops. I was supposed to leave, but not end.
Hmm.
But I made a mistake. I was supposed to leave, but I hit leave and end meeting, so that didn't work. Damn it. Okay. We can continue where we left off. So for nucleotides, do you see how, like, you know most of this stuff, but do you see how the deoxyribose and ribose differ?
Hmm. Yes.
Okay, yeah, the ribose has that extra OH group. And this is just an example of polymerization for
Okay.
nucleotides.
Right.
Make sense?
Yeah.
Okay. Um. So yeah, we know what DNA does. Okay. Now RNA. Now this stuff is, now we're gonna talk about things that matter. At least this one is. So RNA is single-stranded, but they can fold back on themselves to form double-stranded segments. And there are also some viruses that have double-stranded RNA.
Hmm.
And so the functions of RNA, we know about the first function, which is genetic information and transmission. Um, but, oh, and then we know that it can help form structure like ribosomal RNA. But the thing that is like new is that it can work as an enzyme, which is pretty interesting because every enzyme that we know of is just a protein, right? But
Hmm.
RNA can act as an enzyme by itself.
Okay.
Yeah. So I would just
So I
write
should just write down those three things, right?
yeah, here. You should write down um so, like, RNA single strands, they can fold back on themselves to make double strands. And then there's also double-stranded RNA viruses.
RNAs in the strand fold back to form double-stranded.
Oh crap! I cleared. Oh Jesus!
It's okay. I got most of it. But just put the thing on the side that you were putting.
It keeps turning on like my speakers that I turn off when I join the Zoom meeting. But anyway. Okay. So. Alright, now proteins.
Wait.
Sorry?
Back up. Alright, so should I write down any of the functions?
Yeah. So we already know the genetic functions. And then the structure, it contributes to making the structure. For example, rRNA, aka the ribosomal RNA.
Mm-hmm.
And then the special one is that it can act as enzymes, ribozymes.
Okay. RNA functions.
What are you using for your note taking again? Something that only works with Macs?
That what?
So, you're using Notability or something?
Yeah.
Okay. Because that only works with Macs, I think.
No, it should work with everything.
Okay. Because I was thinking, like, I could just send you, like, some of these. But, I don't know, maybe it's better if you draw it yourself. But
I mean, what do you call it? If you send me, like, PDFs and stuff, I can convert them
okay.
on here.
Mm-hmm.
Like, it's a PDF converter. It can convert anything.
Alright.
Yeah. I can convert, like, everything. Okay. Good. Done.
Okay. Now, yeah, proteins. So, they're essentially polymers of 20 different amino acids. Unique arrangement of the amino acids gives them unique shapes, allowing selective interactions. Typical cell may have 10,000 different proteins. They function as pretty much they do like pretty much everything. So this is just an example of, another one of the polymerization reactions, which is, always going to be dehydration synthesis.
Yeah.
And then there's primary structure, secondary structure, and tertiary structure.
Yes. Always manages to come up.
Yeah. So yeah, you could decide if you want to like write it out like how I wrote it out, which is weird for some reason. It's like things have moved.
Okay.
Like, yeah, the primary structure is just the amino acid backbone. Yeah, so like, precise order, yeah. Blah, blah, blah. Held together by peptide bonds, essentially.
Uh-huh.
So, yeah,
Okay.
and then they have side chains, which can determine, if it's acidic or not. So,
Got it.
yeah, side chains, they're not part of the peptide bond. So, like, this is the peptide, backbone, and these are the side chains. Now, secondary structure, essentially gives you an idea of, so spatial organization. So I wrote, like, spatial arrangement of amino acids when they're near one another. So, there's an alpha helix, which I drew over here. You see that?
Mm-hmm.
And it's because of hydrogen bonds forming between, like, an amine group of one amino acid and, like, a carboxylic, carbonyl group, like oxygen on another amino acid. And this is like an overhead view of an alpha helix. So you see how, like, we're looking down it. All these R groups, which are the side groups, the side chain groups, they're on the outside of that helix structure.
Okay.
So here's their drawing of it.
Oh.
And it looks like crap.
It's terrible. Okay, just give me a second here. All right, can you put your notes back up? Because I just wrote the forms here.
Oh, yeah.
They basically do everything and stuff.
Yeah, the other type of secondary structure is a beta pleated sheet.
Okay.
Yeah, and so does this overhead view kind of make sense over here?
Yeah.
Whoa. I think a bird just hit my window. Okay, so yeah, it's like, see if you look down the axis of this helix, that's what that's supposed to be. And see, the R groups are the side chains. Now, if the side chain groups are hydrophobic, then it can do something like this. It can insert itself into a lipid bilayer or cross a lipid bilayer.
Yeah.
Because of the hydrophobic side chains. All right. Now, sometimes 2 or 3 alpha helices can wrap around one another. So, it can do that to minimize exposure to hydrophobic amino acid side chains to, like, an aqueous environment. So, that's called a coiled coil. There's whatever, heptad, etc. Now, beta pleated sheet, so their drawing for that is better, actually, because it's kind of harder to draw like the 3-dimensional version of this.
Mm-hmm.
But in my note, I do have you see how the hydrogen bonds are still being formed between the hydrogen of the amine group and the carbonyl oxygen.
Yeah.
Yeah. So that's what's happening here. Yeah, and so and then they bend. There's like a turn as well. So so you'll start to see like proteins drawn in this type of way. It's like whatever, a ribbon kind of thing. So like essentially like the ribbons indicate beta pleated sheets, and these like like spirals indicate the alpha helices.
Yeah.
All right. Yeah. So like the average polypeptide, 60% of the chain is composed of these secondary structures. And there could be like, for the parts that aren't, there could be hinges, turns, loops, etc. But these, secondary structures are often the most flexible and where the most biological activity occur.
Yeah.
Now, tertiary structure, I wrote as it's a 3D arrangement, and it's mostly held together very loosely by hydrophobic interactions. So I kind of just drew like this over here, but okay. Conformation of the entire protein, non-covalent interactions between R groups. Yeah, so once you get to the tertiary structure, it's like secondary structure is just, it doesn't involve the side chain, the R groups, right? It just involves the backbone. But tertiary structure now involves the side groups. Okay?
Got it.
And
right.
Yeah, and they're going to be like hydrophobic interactions and stuff like that.
Okay.
Okay, so that looks a lot like what I drew over here. Like this thing over here. So like here's an example. Like these are like van der Waals forces. So like this is like the green thing is the overall tertiary structure of the protein, and then it's just showing you the interaction. So there's van der Waals forces between these two like benzene groups and these two methyl groups, these two like whatever, like I think that's like a leucine or something, and there's hydrogen bonds here, and there's ionic bonds as well. All right. Now, yeah, this is an important one. Disulfide bridges. So, because everything that we talked about before were non-covalent, but a covalent bond may form, and it's called a disulfide bridge. And it's because cysteine, which is a amino acid that has a sulfur in it, can react, like it can be like oxidized or reduced or whatever, like these two guys, to connect and to form this disulfide bond.
Got it.
Yeah, so you have polypeptide 1, polypeptide 2, and they can link up as a disulfide bond. So, like when people use like when people chemically straighten their hair,
yeah,
they do that. What they're doing is they're breaking the disulfide bonds in curly hair.
Okay.
Yep. Yeah, here, check this out. So like chemistry of curly hair. Oh, I think they do the reverse actually. So like curly hair. Yeah, I forgot which did which. Like, I forgot which one is responsible for the curls and which one isn't. But essentially, that's disulfide bonds, straight, curly. Okay, straight hair, I believe, has the disulfide bonds. And okay, and then you break them, and I suppose you reorient the strands like this. All right, I get it. So basically, you break the disulfide bonds, and then you orient it like this. If you want to curl it, so that's why they use like the curling iron that has like a cylindrical kind of shape, and then they make like the disulfide bonds again. But anyway, that's just an example. Anyway, disulfide bonds, right?
Mm-hmm.
Okay. All right, we pretty much know this stuff.
All right, so can we just go through the primary through tertiary before we keep going for a bit?
Yeah.
Like on what you have in your notes, so I know what to write down.
Oh, yeah. Yeah, sorry. For some reason, I think like when I was trying to copy and paste this to something else, it got kind of like messed up. But let's see if I can. So like for primary structure, I would write, amino acid backbone, which is made by dipeptide bonds and disulfide bonds. And then here it's kind of hard to see, but I wrote like the double arrow. And it to basically say what's the point of it, and it determines how the protein will fold.
Mm-hmm.
And then for secondary structure, I write the spatial arrangement of amino acids near one another, and then I write these down. I'm sorry. And then I drew out these structures. And then for tertiary structure, I write it's the 3D, three-dimensional arrangement. All right, and it's held together very loosely by like essentially like hydrophobic interactions.
Wait, can you go back up?
Yeah. Where like?
Yeah.
How high?
Of amino. No. Yeah, there. The secondary structure. Determine spatial arrangement.
Yeah, of amino acids near one
one.
another. Mm-hmm.
Near one what?
Oh, sorry, near one another.
Okay.
Yep. And yeah.
Okay.
Yeah. If I were to add something to that, I would say hydrogen bonds between the peptide backbone, not between R groups.
Okay. And then the alpha helices and the beta peptide sheet are like examples?
Yeah.
Now for tertiary?
Yeah, so for tertiary, I wrote 3D arrangement, mostly held together loosely by, I wrote like hydrophobic interactions, but I'm going to add to that. I'll just write intermolecular like forces. And disulfide bridges. What's up?
By hydrophobic?
Oh yeah. I want to like cancel that. Sorry. And I wrote an arrow to be because I to write just intermolecular forces because that includes hydrophobic, that includes hydrogen bonds, that includes van der Waals, because it includes all of that.
Okay.
And then also disulfide bridges, because disulfide bonds are intramolecular, so that's why it's in addition to that. And then I should actually also write it's between the R groups. So secondary structure does not include the R groups, but tertiary involves the R groups.
Okay, so secondary doesn't involve R groups, but tertiary does?
Yes.
Involves R groups. Okay, cool.
Yeah. And then quaternary structure doesn't always exist. It exists when there are subunits.
Okay.
So like so yeah, so that just includes when there's like subunits. So an example that they usually give is like myoglobin and hemoglobin.
Doesn't always exist, right?
Correct, yeah.
Doesn't always exist, then what? Subunits,
Yeah, it involves subunits. So an example that they usually give is myoglobin compared to hemoglobin. So myoglobin is like what's in our muscles, and it's just, it's like it just has like the tertiary structure, but hemoglobin is composed of 4 subunits, and like how the subunits interact with one another is what quaternary structure is.
And the most common examples are myoglobin and hemoglobin?
Yeah, where myoglobin is something that does not have quaternary structure, and hemoglobin does. So they have a image here of what I believe to be hemoglobin.
Okay.
Yeah. So in this picture, I'm pretty sure this is hemoglobin. So see, there's an alpha subunit here, beta here, beta here. So this has 4 subunits. And the way that the subunits interact with each other gives it a quaternary structure.
Okay.
Yeah. Make sense?
Yes.
All right. Now, let's see. Not all proteins assume. Okay. All right. So essentially, what happens with proteins is that they naturally form those. They naturally fold to make the secondary structure and tertiary structure and stuff. But sometimes that doesn't happen. So they need chaperones, like chaperonins, to help them do that, to fold correctly. So here's a nearly synthesized, partially folded protein, and if it's left to its own devices, it'll be incorrectly folded. But if it has chaperone proteins, it's going to help fold it in a correct way. Okay?
Okay. So hold on. Chaperone.
Mm-hmm.
You sometimes use chaperone protein?
Yeah. If it can't fold correctly by itself.
And help them fold if they can't fold themselves. Okay, cool.
Yep. Okay, now there's something called protein domains.
Mm-hmm.
And essentially what that means is that they essentially just have different functions, like independent functions. Yeah. Like usually you'll see it as like, for like an enzyme, like you could say like, this domain is responsible for, is like where the active site is, like where the substrate binds to.
Yeah.
So yeah, I would summarize this by saying domains have, okay. Have independent functions and shapes. So, you got that?
So protein domains are independent, have independent functions and shapes, right?
Yeah.
Okay.
Okay. Now, how proteins are controlled essentially is by altering the shape of the protein. So, we call it allosteric, which means that it can change shapes, and that's regulated by, maybe something can bind to it or something can't, right?
Mm-hmm.
So I would write something like this. Shape, the arrow function. Essentially, the shape dictates the function. They're essentially one and the same. Oh, and then I put that under allosteric.
Protein shape equals arrow
Allosteric.
function. And together. Cool.
Okay, so this is an example of something called phosphorylation.
Mm-hmm.
And essentially, like a side chain of either serine, threonine, or tyrosine, they all have in common an OH group.
Mm-hmm.
And OH group, think nucleophilic. So it attacks the phosphate and it makes a phosphate add to that amino acid. So that is how a protein can be phosphorylated. It happens like all the time in biochem through like something called a kinase, essentially. So things can turn on or off based on a gain or loss of a phosphate group. Yeah, here's like another example. And then here's kind of like an example of like, this is like dynin and kinesins and stuff like that. But it's essentially just like, kind of like the myosin and actin kind of thing, where like it being phosphorylated makes like a bond happen. So this thing like binds to that, and then one thing like phosphate is like lost, so then this thing leaves, and then it like essentially helps it walk along. And yeah, that's it for this PowerPoint. Now I'm going to try to do what I did last time by leaving and then coming back. Let's see if I could do that. There's an end button. All right, now I'm going to hit leave meeting, and you stay in here. All right, okay, now all right, that worked. All right, that worked. Now we're back. I didn't have to make a new one.
Awesome.
All right, are you ready for this one?
Yes.
Okay. DNA and chromosomes. So
wait, just to finish off the last one, anything worth noting towards the end?
Oh, just essentially that phosphorylation can affect the activity of a protein.
Phosphorylation affects activity of a protein?
Yeah.
That's it?
Yeah. They have like specific examples, but we don't really need to know the specifics now. And if we do need to know the specifics, I would tell you like something like even more specific. Like for this one, I can talk about like exactly what's happening, but yeah, we don't have to go that specific right now. Not at least not in your notes.
Got it.
All right. Are you ready for this one?
Almost.
Okay.
Okay.
All right. So DNA and chromosomes. So this is so freaking annoying, man. Yeah, man, sorry. I thought that was going to fix it, but I think it's because I'm logged in. There's 3 people technically, so I'm just not going to use my tablet. So, apologies for that. It should be working fine now. And, yep. Tell me when you're ready.
I am. Alright.
Okay.
I thought we left this shit already.
So, okay, this is pretty cool. So this kind of talks about, like, the major, like, kind of discoveries in this, because Mendel was the first person who essentially discovered how inheritance works. And, like, later on, they discovered these things called chromosomes. And then they discovered homologous chromosomes, and followed by the crossing over involved in, chromosomes. And then Morgan determined that genes could be mapped along the length of a chromosome. And then they discovered that DNA was the genetic material. And then the last thing was the structure of DNA. Now, why do you think they discovered, if DNA is like where it all starts, right? Why did they discover that last?
Well, technically, well, to know DNA structure, wouldn't they need to, like, what do you call it? Know, like, the whole mapping of it, regardless?
I mean, like, they discovered mapping before it. But I mean, just think about it. In 1860...
Oh, wait, there was no microscope, huh?
Yeah, so that's why, so, like...
I was thinking too hard.
So it has to do with that, but, like, how are they able to find chromosomes? Now, that's because using a regular light microscope, you can still see chromosomes. Like, during metaphase.
Yeah, but you can't see anything else in an electron after that, right?
Yeah, exactly. It's because DNA is so tiny. So that's why, they discovered it very, like, at the end.
Yeah.
And then the structure of it was from, like, X-ray crystallography.
Got it.
Yeah. So, like, they basically knew about inheritance for, a long time, and as we learn more and more about it with improvements in technology, we can see the tinier, smaller units.
Got it.
Yeah. Now, yet again, building blocks of DNA, right? Sugar, phosphate, blah, 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 the base, etc. So, that's the sugar group, the phosphate group, which makes that backbone, right?
Yeah.
The phosphate and the sugar make the backbone. And then the base, the nucleotide, right? And then, these are the different ones. Yeah, and then there's a 5' end and a 3' end. And then, the bond between the two, it's called a phosphodiester bond.
Yeah.
Yeah, and that's because it's, yeah. And, okay. Yeah, and now, they're going to be complementary, anti-parallel. So, you know that stuff, right?
Yeah.
Okay. Spiral staircase. Double helix. Major groove, minor groove. Yet again, DNA structure. So a chromosome is a long strand of DNA. So remember when it, like, condenses with histones and stuff?
Yeah.
It can be, like, visible. Okay. So yeah, we can notice like there's locations of genes on that chromosome. And then yeah, essentially a gene is just something that, gets transcribed into RNA, which then gets translated into protein.
Yeah.
And yeah, they're arranged along the chromosomes. So, essentially, we have, this like double bond over here. Double strand over here. So this entire thing is a segment of a double-stranded DNA, comprising 0.5% of the DNA. What the hell? Hello?
Hi, is Rita there?
No, why?
Oh, we were calling for either one of you. This is Bill calling for support our police. Our mission is to seek police training
One sec.
improvement, as well as to advocate for a death and disability fund for those officers killed or injured on the job with the passage of better laws. As a PAC, we stand with Representatives Abigail Spanberger and John Rutherford for supporting law enforcement. Help us elect the people that will back the men and women in blue and effectively handle these issues. If we were to send you an envelope, would you show your support with a one-time donation?
I don't think I can. I'm sorry. I'm in the middle of something right now. Can do this some other time?
I'm sorry.
Thank you.
Thank you for your time.
Sorry about that.
No problem.
It's like some scam likely thing and someone wanting donations. Anyway, so this entire thing is part of like this yeast genome, and essentially the genes are the yellow things. And all the other stuff aren't genes, and this kind of like is what we talked about like a while ago when we, in your other class, bio class, where like 1 to 2% of the genome actually codes for things.
Yeah.
So yeah, it says note spaces between the genes. At any one point on the chromosome, why is only one strand yellow? That's a pretty good question. Like, for instance, like they don't overlap here, right? So why do you think?
Well, they're supposed to be. Well, I mean, this is compromising. Wait, what? Hold on. Any one point on the chromosome, why is only one strand yellow? Because that might be a different gene that it codes for something else.
Well, then it would still be a gene, right? And it'll be yellow.
Oh,
but
okay.
But yeah, it's because essentially, like, when DNA is transcribed, like the double helix, the two strands separate, right?
Oh, because there's a separation between the strands, is what it is.
Yeah. So when they separate, right? Only one of when they separate, one is like a template strand and one is a coding strand. So when they separate, on the strand that's there for the gene, it's going to, transcribe that into RNA.
Got it.
Oh, they don't even explain it. But essentially, like, if I were to draw it, like, we have two strands of DNA. And let's separate them. And then for our RNA, wait, let me do this. Like 5 prime, 3 prime, 5 prime, 3 prime, right? Now 5 prime, 3 prime, 3 prime, 5 prime. And now for the RNA, right?
Yeah, okay. I get it now.
Yeah, so RNA goes like 5 to 3. So that is not going to work. But this one on the right is. And so essentially what that means is, so like let's say the gene is here, right?
Mm-hmm.
Then that means here is the complement for that gene. Right?
Got it.
Like if this is A T, this would be TA, right?
Mm-hmm.
So this part, that's TA. When RNA like polymerizes along this template strand, the RNA is going to make AT. But not the T part, but like U or whatever.
Mm-hmm.
But that's how it'll make, the gene that will actually be translated.
Got it.
Yeah. So like this is called the coding strand, and this is called the template strand. And the coding strand is responsible for having the actual code for the gene, and the template is a complement of that. And when you actually transcribe that into RNA, you need the template strand because it's going to make an RNA sequence that's going to be identical to the coding strand, but it's going to require the template strand. That's why they don't overlap here. Right? Because this is a gene over here, then this over here is the complement to that gene.
Uh-huh.
Yeah. So that's the answer to that question.
Got it.
Yep. Okay, now genes are the unit of inheritance. Chromosomes physically carry the genes. Alright, an allele has whatever, 2 copies of a gene, 1 from each parent. Dominant, recessive, yeah, you know that stuff. Homologous pair of chromosomes. So, flower color gene, locus is a location, right? You good with that?
Mm-hmm.
All right. So
Wait, say it one more time. You broke up.
Oh yeah. Oh no, I was saying, are you good with this?
The homologous chromosomes.
Yeah. Like the locus, which is a location for the gene that determines flower color, is here, and one one of your parents may have an allele for purple flowers. Your other parent maybe for white flowers, but yeah, they're homologous here.
Got it.
Yeah. This is just where they use fluorescent probes to be able to identify.
Yeah.
Abnormal chromosomes. Are associated with inherited genetic defects and many cancers. So we have chromosome 6 here and chromosome 4 here. And here we have a type of chromosomal mutation, which is the worst type of mutation because a DNA mutation is on a small scale. But if you have a mutation at the chromosomal level, it's huge, right?
Yeah.
So what you basically have here is like a translocation, like a reciprocal chromosomal translocation. Like essentially the bottom half of this goes to chromosome 4.
Mm-hmm.
And then the bottom half of this goes to chromosome 6, so that's why they kind of split. It's like a reciprocal here.
Mm-hmm.
Yeah. Replication and segregation of chromosomes occurs in the cell cycle of proliferating cells. So, you remember this thing? Interphase, mitosis, cell division, etc.
Mm-hmm.
All right, 3 DNA sequences needed to produce a chromosome that can be replicated and segregated in mitosis. Origins for initiation of DNA replication, centromere, and the ends, which are the telomeres.
Yeah.
So,
Yeah, the meeting points, then the telomeres, and then, yeah.
Yep.
Okay.
Yeah. The origin of replication is where, it starts to replicate, so it creates that bubble.
Yeah.
And these are the
And then
new
the centromeres
strands.
are in obviously the center.
Yeah.
That brings the telomeres together and everything.
Wait, sorry?
It's the centers that bring like the, what do you call it, things together, right? Not telomeres, because the telomeres are at the end of that bubble.
Wait, so like this thing opens up and see this over here and that? Those are the new ones being replicated, right?
Yeah.
And, they're going in opposite directions. Like, if I have 5, 3, 5, 3 like that,
Mm-hmm.
and, they separate, then the new strand will go, like, 5, 3 in this direction, and then this new strand, 5, 3 would go in that direction, right? So overall, this replication bubble will involve, a new one being kind of synthesized in this direction. So, yeah, wait, sorry. If this is, 5, 3, and this is 5, 3, like that.
Mm-hmm.
This new strand is going in the 5. So, remember, it has to be synthesized in a 5 to 3 direction.
Mm-hmm.
So, we have, a double strand, 5, 3, complementary 3, 5. And so, the 3, 5 one, it has to synthesize this new strand in a 5, 3 direction. And since, this is a 3 over here and a 5 over here, this one will synthesize the new strand in that. It still has to be 5, 3, but it'll be going, in that direction. This one's going here in this direction, this one's going in that direction. Right?
Mm-hmm.
Yeah, so if this is 5 and that's 3, right? Then this over here will have to be 5 here and 3 here and synthesize up. And then this one over here will be 3 here and 5 here and synthesize going down. Okay?
Got it.
Yeah. So, yeah, that's the origin of replication, and this is the replication bubble. And, this is the centromere. And this is another origin of replication, so, that's being replicated as well. And the blue stuff are at the ends. Those are telomeres. And they basically just don't have anything that codes for stuff. They're just a bunch of copies. And, they just are there to make it. Because every time we replicate, the entire DNA gets shorter. So yeah, anyway. That's why in this step, you now have... you go from having one double strand to 2 double strands, right? Because of that this replication phase. Yeah. Oh yeah, and then the mitotic spindle attaches to the centromeres, and yeah, that's basically that, right?
Yeah.
Okay. chromosome, centromere, chromatid. All right. Now we have higher order repeat, centromere DNA. Okay, this entire thing is a centromere DNA. Alpha satellite DNA monomer. Okay. All right, so this alpha satellite DNA portion is what the it binds to kinetochore proteins.
Mm-hmm.
So the kinetochore thing binds to the alpha satellite DNA monomers, and then the kinetochore is attached to the spindle apparatus. So,
Yeah.
Those are at opposite ends. Yeah. Interphase chromosomes are organized in the nucleus. Okay. Yeah, and you remember, like, this stuff with, like, the histones and things like that?
Mm-hmm.
Yeah, that pretty much determines, like, how much it condenses and how much is expressed. So, yeah. So chromatin is essentially a name for DNA and proteins. And histones are proteins that have a lot of basic amino acids. Now, this is worth writing down that basic amino acids are positively charged, in whatever the cell. And we know that DNA is negatively charged. That's why they, bind together.
Okay.
Yeah. So, once they bind together, we call it a nucleosome.
Okay.
Yeah. Now you see here, like it's DNA here, and then here they call it beads on a string. So the yellow thing here is the histone, and it loops around it, and then here it's just like regular DNA, and then more of these histone, like nucleosomes essentially. And with enough of them, it's going to be like compacted. It's going to get like closer and closer together. Mm-hmm. And these things fold into loops, and in its most condensed state, it'll make a chromosome. So that's why a chromosome, is like that. Like we can see it because it's really compacted. So they say that the chromosome is 10,000 times shorter than the entire length of the DNA if you unraveled it.
Got it.
Yeah. Now, there's also the thing about whether it's transcribed or not, because you need see this area that can be transcribed and expressed, but the more you condense it, the less it's going to be able to be transcribed and expressed. So,
Yeah.
yeah.
Got it.
Okay. DNA packing and gene expression. So they're going to talk about that. Heterochromatin, regions that do not uncoil after mitosis. So, okay, this is useful to write down the difference between euchromatin and heterochromatin. I would add to that, heterochromatin is essentially, tightly wound, which means that it is not expressed. Oh, I think we might be almost out of time.
Mm-hmm.
We started at 3:20.
Yeah.
Okay, so we'll do a couple of more minutes. I got all this new furniture that I have to build.
Okay.
All right, so you wrote euchromatin loose and expressed, right?
Mm-hmm.
Tightly, okay. Chromatin structure varies along a single, okay, interphase chromosome. Yeah, so centromeres and telomeres, since they're not expressed, they're going to be heterochromatin. And you might have some bits in between, but yeah, they vary along, So you'll get some euchromatin and stuff. Okay, now, and then here's kind of going into it. If we have this chromosome and we take this small region, and they say this part is heterochromatin, so that's not going to be expressed, so we don't have to worry about that. But yeah, if they take this small region and they zoom in on that, they get, so that's 10% of this chromosome arm, which corresponds to 40 genes. So each of these things are genes.
Mm-hmm.
And then, so now they're going to, focus in on just this area over here. 10% of the 10%. So 1% of the chromosome. And that just zooms in on this. So that has 4 genes. Okay. Now, if we zoom in on one of the genes, right, we get, the actual DNA portion. So that's why you see the double strands. And so we have regulatory DNA sequences. So that, means promoter, whatever, that kind of stuff. And then, the exons, the introns, right? And then, it gets expressed, turns into the protein. Right? Makes sense?
Mm-hmm.
All right. Oh, that's it for that.
Yeah. It was a short one.
Yeah.
I was just reviewing that.
Mm-hmm.
All right. Well, then that concludes the time, basically, right on time.
Yeah. Okay.
All right, then. So, take care, man.
All right. You too, man. I'll see you next week. I'm gonna build some of that furniture now. All right.
Awesome.
Take care, dude.
All right. Take care.
Yeah.
Bye-bye.